Hello, everyone, and welcome to today's webinar, Validating Single-Cell RNA-seq Data Using Single-Cell Western Blotting and RNAscope Technology. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Advanced Cell Diagnostics. To learn more about our sponsor, please visit www.acdbio.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Brian P. Hermann, the Associate Professor at the University of Texas in San Antonio. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Hermann, you may now begin your presentation.
All right. Thank you very much, everyone, for tuning in. Again, my name is Brian Hermann. I'm from the University of Texas at San Antonio. My laboratory is primarily interested in studies of spermatogenesis, which is the process of sperm production in the testis. And in particular, we study the stem cells that form the foundation of the spermatogenic lineage. And as a part of our work, we have done a considerable amount of single-cell gene expression analyses to begin to understand more about how those cells function and how they're related to other cells within the germ cell lineage in the testis. And consequently, we have been responsible for validating our single-cell data. And so I'll tell you a little bit about what my lab has been doing at the single-cell level and how we've been approaching the validation problem, both in our published work and unpublished work that I'll share with you today using the Milo and RNAscope approaches to validate our gene expression results. So first, just to give those of you who may be unfamiliar with the testis a little bit of a primer on spermatogenesis. This process occurs, of course, within the testis, and in particular within structures that are called seminiferous tubules. These are tube-like structures that are repeating, and several of them are located within the testis. And within each seminiferous tubule, you'll find that the most primitive germ cells are located closest to the basement membrane on the periphery of the seminiferous tubules, and as germ cell maturation proceeds, those cells move closer and closer to the lumen, where ultimately they're released as mature sperm. And so you can see in this particular cartoon that illustrates a cross-section through a seminiferous tubule, the complexity of different cell types that are found in any particular segment of a seminiferous tubule. And so this really underscores sort of the experimental problem that we in the spermatogenesis field are faced with, which is how to pin down the biology of individual cells that are participating in this lineage when the complexity of the tissue is so high. And so to give you an even greater understanding of the complexity, if we focus exclusively on the germ cells, we can see that underlying the spermatogenic lineage is, as I mentioned earlier, the stem cell that forms this foundation and enables germ cells to be produced throughout the lifespan of adult males. And so we call this cell the spermatogonial stem cell, and it can self-renew or initiate differentiation to produce what I call the rest of the spermatogenic lineage, but cells that will proceed through mitotic amplification, enter and proceed through meiosis, complete the second meiotic division, undergo morphological differentiation to ultimately produce sperm that you can recognize as compact nuclei with a flagellum. And so this system is awesome, in my opinion, because it gives us the opportunity to peek into the process of meiosis, cellular differentiation, stem cell fate, and has really strong implications for understanding of normal fertility, pathology of infertility, how we can interfere with fertility and contraception, stem cell transplantation, in vitro culture systems for making gametes, and really there are many, many, many more. So we've been very interested in understanding how the stem cells function, how they're related to their most immediate progeny that we call progenitors, and really how the process of spermatogenesis is regulated. And we have a lot of holes in our understanding, so there's a really ripe opportunity for expanding our knowledge. But there's a problem, and that is, you can see in this diagram that there are many different cell types that we can recognize based on decades of experimentation in the testis. But our ability to separate those cells is really limited. So there are methods available that will allow us to select spermatogonia that are mitotic or meiotic spermatocytes around spermatids, and there are markers that can allow us to select some subgroups of these. But what you'll see is that our ability to resolve individual cell types is pretty limited. And so this limitation means that we're going to necessarily aggregate varieties of cells when we do molecular profiling. So this aggregation of molecular analyses really poses some important biological implications. So consider that a population of cells may have a gene expression pattern that's indicated by the colors of the cells shown on the screen. So blue cells have relatively low amounts of expression of any particular gene, and the yellow and red cells may have higher levels. But if you aggregate those cells, the result is that you get a population level result that constitutes the average across the entire population. And so when you look at the average, as shown by the green line in this plot below for the gene expression signature of that particular population, the result doesn't match at all either of the major populations found in the bulk. And another example would be for a relatively rare population of cells, the results from those particular cells will be diluted out by the majority, and so the bulk average looks exactly unlike the cells of interest. So this challenge really serves as the foundation for needing an ability to assess gene expression at the individual cell level. So the solution that my lab has taken to this problem of aggregation, as well as a number of others in the field of spermatogenesis and many other fields, is to look at gene expression at the individual cell level across their entire transcriptome. So we have isolated testicular biopsies from human patients as well as adult mice and immature mice, and isolated cells from that testicular tissue, including unselected populations that include all of the cells within the testis, as well as various sorted cell populations that enrich for particular cell types, and then profiled these cells using single-cell RNA sequencing, using a couple of different commercial platforms, including the 10x Genomics and Fluidigm C1 platforms, and then use sophisticated data analysis pipelines to establish clusters of cells that differ in their gene expression profiles and model those differences and developmental relationships in pseudotime, and then establish gene expression signatures that show how those cells change as they proceed through development. And this has really opened up a number of different avenues of investigation and expanded our knowledge in how spermatogenesis works by considering all the individual cells instead of simply examining groups of cells in aggregate. But these And so to give you an idea of the kind of results that we have obtained, here is a tsne projection showing all of the cells that we extracted from the adult mouse testis. So each dot on this plot represents an individual cell, and you'll see that the dots themselves are not located all in the same spot. They're scattered around the plot, and that shows that there are differences in their individual cell transcriptomes, but they're also grouped by similarity, and that's what establishes the coloring, which reflects how similar those cells are to each other. So individual clusters are colored similarly, and so we can establish what are the gene expression differences between individual clusters, and that's shown in this heat map that just appeared to the right. That shows the top 10 differentially expressed genes between clusters, and this is kind of meaningless unless you look at what the individual genes are, but if we look at some specific genes that we know mark particular cell types, we can then establish which clusters of cells reflect which cells in the testis, and that's shown in this diagram at the bottom, where I've aligned different clusters, for instance, cluster 16, with particular cell types. So cluster 16 contains undifferentiated spermatogonial, some of the earliest differentiating. So this includes the stem cell population. So this has allowed us to really understand how germ cells within the adult mouse testis, in this case, are changing in their gene expression patterns across that developmental lineage. But these results really assume that the data that we've generated reflect bona fide single-cell transcriptomes and that the data aren't artifactual in any way. And so we've spent a lot of time, and others have as well, validating our single-cell transcriptomes. And so, for instance, we've performed studies where we mix cells from different species, in this case from human and mice, together before performing our single-cell profiling, and asked how often do reads from mouse and human co-localize, which could only occur if we were detecting expression from a multiplet that contained a droplet or such that had more than one cell, one from each species. And you can see that there's relatively infrequent multiplets. But also, then, do the transcriptome results, for instance, the expression profile of particular genes, match the expression profile that we have already known in the literature, or which we can detect using antibodies. So shown on the right are the profiles for 3 genes, DDX4, which marks germ cells, GATA4, which is a general somatic marker, and KCTD9, which appears to be expressed in spermatocytes. And you'll notice that for each of these genes, there are multiple histograms that we call violin plots that reflect the expression level among cells within that particular population. So, for instance, GATA4 is essentially absent except for cells within the last 2 clusters, clusters 15 and 17, which are somatic cells, while DDX4 is very highly expressed in all of the other clusters, and there's a little bit of boil over into the somatic cell cluster. So we took these data and extended them back in situ into the testis and used immunostaining with antibodies against GATA4, DDX4, KCTD9, and a related marker called TRA98, which is another germ cell marker, to validate that the protein was found in the same cell population situ that we detected the RNA for those markers in the single cell studies. And the long and the short of it is that the results generally match up. So our expectation that, for instance, KCTD9 is expressed by spermatocytes matches the detection of the gene shown in red, the gene product KCTD9 protein, is most abundant among spermatocytes, which are one layer off the base of the seminar. So this is a great way to validate that the results are telling us something important. But the limitation is that this is extremely time-consuming. It's low dimensionality and really leaves a lot of information uninterrogated on the table. So we've also extended our validation to a higher dimensionality by validating these gene expression signatures at the RNA level, using, in this case, bulk samples of intact testes from mice as the first spermatogenic wave proceeds during postnatal testis development. The beauty of that system is that each population of cells sequentially emerges in development, so we can detect the signature associated with a new population as it emerges, and then go back and correlate that with our results that we obtained in our single cell analysis. So, for example, here are qPCR data from testes that were obtained from mice from postnatal day 6 through postnatal day 30. And shown in this heat map is the levels, relative levels of RNA for a panel of 33 genes that mark discrete cell types in the adult testis based on our single cell data. And you can see that at postnatal day 6, the first 3 groups of genes, which are color-coded to match the scheme that I've shown on previous slides, are already present, and so their levels are already abundant, but subsequent populations of cells begin to emerge sequentially as you move from left to right across this heat map, and that reflects their initial appearance during the first spermatogenic wave. So this allows us to then correlate the signatures that we detected in single cell at the RNA level with the same expression of those genes during the first wave of spermatogenesis. But the problem is this has lost our singularity, so we don't necessarily know what cells are expressing these genes because we've analyzed the results in bulk. So really what this means is our primary methods of validation both come with some compromises or really limitations, I would say. So protein level validation with immunofluorescence or indirect immunofluorescence means that we retain the singularity. We can see the detection of the protein at the single cell level in situ, but we lose a lot of the quantification ability. It becomes semi-quantitative at best, and the throughput is considerably low, down to 2, 3, or 4 markers at a time. And really the assumption is that there's a congruency between the RNA detection that we've detected at single cell level and the protein that we're detecting in situ. And that may or may not be accurate based on the level of post-transcriptional regulation. This is a particular problem for us in the germline where there's a considerable amount of post-transcriptional gene regulation where particular gene products may be sequestered and degraded or preferentially translated or not. So, and in addition, of course, the assumption is there are antibodies that are validated that are available to detect the gene product of interest. And many of the folks in the audience, I'm sure, will laugh when they test an antibody and maybe it works and maybe it doesn't. And that's not a trivial undertaking in a lot of cases. And RNA level validation, at least the work we've done in bulk, retains the quantitative information, but and the throughput is really instrument dependent. It can be a few genes or hundreds of genes depending on the kind of technology you have, but necessarily you lose the singularity. So none of these is perfect, and really, can we validate single cell data without these compromises? And so that's the question that my lab has been faced with and has used now a couple of different technologies available from Biotechne to address this problem. The first is using Milo, which is a single cell Western Blot system, which allows profiling of thousands of individual cells at the protein level for up to 12 proteins per cell in a relatively short amount of time, within a day, using antibodies that are off the shelf and validated for Western Blot. And to give you a sense for how this works, You begin with a suspension of cells, load them into a kind of slide-like chip that has a thin polyacrylamide gel layered on top, and then load the cells onto microwells within that gel by gravity settling. And really, this means that by Poisson distribution, you'll fill a subset of the available wells within a particular chip. And this is dependent on how many cells you load and how long you allow them to settle. And then lyse the cells within the well and electrophoresis the products in one direction into that thin polyacrylamide gel, and then immobilize the products with UV light, which then allows you to perform in-gel Western blotting with antibodies of interest that you have maybe validated with a standard bulk or aggregate approach, and then use secondary antibodies that are conjugated to fluorophores so that the gels can then be imaged with fluorescence based techniques. So really, in a matter of just a few hours, you can get at single cell resolution Western blot data on particular gene products of interest in thousands of individual cells, and this is something that is facilitated by the Milo and a microarray chip reader. So to give you a sense for what the data look like, here is a scanned chip that came straight off the microarray scanner from a Milo experiment, and you'll notice that there are lots of dots that appear in rows, and in a subset of those dots, you'll notice that there are white splotches, and those reflect the fluorescent signal from the Western blotting of the proteins of interest. But then a lot of those wells don't have any or didn't have any cells, and so there's no detection of proteins in the lanes below. But for each lane, the amount of signal can be quantified, so the area under the curve can be established and then plotted according to the distance from the center of the well, and different products can be co-quantified in the same sample. So in this case, I'm showing you detection of GFP from GFP-labeled cells, as well as an internal control with an antibody against histone H3, which should be present in all cells in this sample. So we used some of our germ cells to begin understanding how well this technology worked in the male germline. And for that, we sorted germ cells that had a GFP fluorescent reporter and put those cells on a Milo chip, and then detected a germ cell marker, which is DDX4, one that you've heard of before. And so shown here are 3 lanes of a gel that have DDX4 positive cells located in them, and these are detected in the Alexaflor 555 channel. And at the same time, we also, in a different channel, detected 2 different proteins, histone H3, which is our internal control, which is on the bottom of this set of wells, as well as TRON98, which is another germ cell specific marker. And so you can see that both of these proteins were detected in the DDX4 positive cells. And we can detect TRON98 and histone H3 together because they resolve at different molecular weights in this gel. And since we have thousands of individual cells, I'm just showing you 3, we can quantify the degree of overlap of these markers. And so shown on the right are some tables that show that the degree of overlap between TRON98 and DDX4. So among DDX4 positive cells, 90% or give or take are, actually more than 90% are also TRON98 positive, but surprisingly, a subset of the the TRON98 positive cells are DDX4 negative. Now, this isn't actually surprising because we do know that there's not complete overlap between TRA98 and DDX4. And so really this shows us that there is both congruency and heterogeneity in expression of these genes. I also mentioned that we had sorted GFP positive cells, so overlap between GFP and DDX4 is shown here, and you'll notice that we didn't do a particularly great job at sorting the GFP positive cells because almost or more than 2/3 of those cells were GFP negative, as shown on the second row. But among DDX4 positive cells, we do detect a large proportion that are also GFP positive cells. Surprisingly, about 1/3 of the GFP positive cells were negative for DDX4. This was something we didn't expect, and is something that we're currently following up on to understand the implications of. So, shown on this plot is a diagram of the fluorescence intensity for one particular well that was marked with 3 different proteins, but that's one cell. And so how can we understand the degree of fluorescence and how it varies across the population? And the beauty of Milo and the analysis pipeline is that it provides the fluorescence readout as a degree of intensity of the protein and the quantification of the protein across your population. So you'll see that there's a log 10 difference in the degree of fluorescence intensity of the DDX4 positive cells, and the same thing is true for TRA98, and an even greater variation in the level of histone H3. So really, this technology arms you with a lot of information about the degree of variation among your individual cells of interest, and could help better understand the underlying biology that's driving your system. And for us, has been very informative in terms of to what extent variations in the RNA then correlate with variations in protein level, which is important in the germline, as I mentioned earlier. And I should give a shout out to Eric Jabart, who was very helpful in doing or helping us with the analysis from this particular experiment. So Milo provides some really essential information at the protein level that allows us to quantify the degree of variation among individual cells and at the single cell level. But as I mentioned earlier, we're very interested in how the tissue is organized. And so the spatial relationship of cells that have different levels of these gene products is also important to us. And that's why we started our validation schemes with immunohistochemistry. So these are plots that you've seen before that show 3 genes, in this case ID4, DDX4 that you've heard of already, and SOX9. SOX9 is a marker of Sertoli cells, which are the major somatic cell type within the seminiferous tubules of the testis. ID4 is one of our favorite markers of the stem cell population in the testis. It also marks spermatocytes as they're going through meiosis. But we're interested in the expression in Sertoli the stem cell compartment. But the problem is we don't have a good antibody for ID4. And so interrogating this gene at the protein level is problematic. We can do this for other proteins like SOX9 and DDX4 and correlate their expression, but with the mRNA levels, and establish their profiles spatially within the testis. But we can't if there's no antibody available, or if the protein is not conducive to detection in that method. So this really is a limitation of the available in situ technologies. So we've instead turned to an alternative, and this is just a rehash of what I just said. we've turned to alternatives, which is to back up to the level of the RNA and see if we can measure spatially the expression profile of our genes of interest. And for that, we have settled on using RNAscope, which is also available from Bio-Techne through their subsidiary ACD. And RNAscope is a technology that has been around for several years now that allows really robust detection of RNAs at the single cell level and at the single molecule level, so that the spatial location and levels of expression of a gene product can be measured. And so their technology is based on a double Z target probe methodology, single signal amplification, and I'll go over this in more detail in some subsequent slides, and then quantification either with fluorescence or chromogenic approaches, and that allows the expression levels of the genes of interest to be quantified in a spatial manner. And this is really, as I mentioned, a really robust technology that allows mRNAs, long non-coding RNAs, and even splice variants of genes to be detected in situ, and is suitable for a wide range of sample types, from formalin-fixed tissues to frozen sections to cultured cells, you name it, and it will work. So the way this works is the tissue or cells are permeabilized. Obviously, you need to be able to get the reagents into the cells. So this is not compatible with live cell detection. So the cells have to be fixed in some way or holes poked in them. And then the target probes, in situ probes, are hybridized to RNA within your sample. That signal is then amplified and subsequently visualized and quantified. And the foundational technology for this approach is this double Z probe design, where you have two independent probes that are structured in this Z-like way, each of which binds adjacent to each other on the target RNA within a 50 base region. So you have two adjacent probes that are binding to your mRNA, and onto that set of probes, the pre-amplifier will then subsequently be able to bind. And so along any given RNA, you have the ability to hybridize multiple probes, and the typical number of double Z pairs is 20. So they have 15, 18, or 20 different sets of probes that will bind at different positions along the RNA of interest. And each probe then will allow the preamplifier to bind to that double Z, and then allow the amplifier to interact, which then allows the either chromogen enzyme or fluorophore to bind to that probe, allowing you to get highly sensitive detection of that mRNA. And importantly, if both Zs don't bind, so each of the 25 mers of that 50 base region doesn't bind, then you won't get the preamplifier and amplification. So the signal will essentially be nil. So that establishes the specificity of this technology. And so RNAscope can facilitate 4-plex manual fluorescence detection or 2-plex chromogenic detection, or up to 12-plex fluorescence detection with a serial redetection method that I won't talk about at all today. And I also won't talk about BaseScope, which is a technology that allows you to visualize very small differences in the mRNA sequence, like at junctions or splice variants and such. But what I will tell you about is our experience using RNAscope with the manual fluorescence assay. And so I'll first show you some of our results with the most important part, which is our sets of controls. So each of the RNAscope assays comes available with positive controls, which are probe sets that recognize what you might consider housekeeping genes. So in this case, RNA polymerase II, peptidyl isomerase B, and ubiquitin C. And you'll notice in the upper left, I have some violin plots that show the expression profiles for these three genes among our single cells. So you see that they're widely expressed, but not uniformly expressed among all cells, which is not uncommon in the testis. So the spermatogenic lineage often does things different than regular somatic cells. But when we look in cross-section through a segment of testis, you can see in this case there's a seminiferous tubule in the middle that is somewhat ovoid, so a little bit acircular, but we can detect RNA polymerase II, PPIB, and ubiquitin C in a good proportion of the cells within that seminiferous tubule. And if we were to zoom in, I'm sorry I don't have a higher magnification, you would see that these genes are detected at single molecule resolution. You can kind of see some of the speckledy labeling for RNA pol2, but for really highly abundant genes, the dots are so abundant that they all merge together. So a great example is ubiquitin C, where it looks like a hazy red label. So this gave us a confidence that our tissue was of high quality, and we're able to at least detect these abundant and widely expressed transcripts. So is this simply background? Well, that was our next undertaking using our negative controls. And for that, we use a probe set that recognizes an irrelevant gene, the E. coli DAP-B gene, which is not expressed in eukaryotic cells. And so in this case, the labeling is essentially nil. There's some background autofluorescence in the testis that we can pick up primarily in the white and green channels in this case, but is not consistent with single molecule fish labeling, as we saw in the previous slide. So really the specificity of this technique appears to be pretty high, at least with these positive and negative controls. So the proof of principle, though, was whether we could get cell-specific detection of genes that we know are expressed in particular types of cells within the testis. And so we went back to that panel of genes that I showed earlier, where we could label for some of them at the protein level, SOX9 and DDX4, but not ID4 because we lacked a good antibody. So in this case, we've detected these three genes at the RNA level using RNAscope, so in situ hybridization for ID4, which we expected to be expressed based on our single cell results in spermatocytes off the basement membrane. And so the probe in white in the panels all the way to the right show the dots corresponding to ID4, and you can see that those cells are one layer of cells removed from the basement membrane. And we do detect, I don't have a picture to show that here, we do detect ID4 in very rare cells on the basement membrane, which are cells of interest, the spermatogonial stem cells. And importantly, we also see that localization of DDX4, our germ cell marker, which is most abundantly expressed in the meiotic cell population, but also detectable in spermatogonia, is where it's supposed to be, as well as SOX9. SOX9 is the Sertoli cell marker, and we see transcripts detected in Sertoli cell cytoplasm, which weaves in and out between adjacent germ cells. And so pattern of labeling is very reminiscent of the pattern of Sertoli cell cytoplasm, which is very nice to see. So we've established the proof of principle. This technique seems to work. So now the acid test was to look for a gene that we have just recently discovered as being expressed exclusively in the stem cell population, and also for which no good antibodies are available. And so unfortunately, because of publication issues, I'm not able to share the gene name with you today, but this gene, gene A, we found was exclusively expressed by a population of cells that we think is the stem cells, and you can see the profile of mRNA at the bottom right in the violin plot. It's expressed by a really small population of cells in cluster 16, and other data shows that that's the stem cell population. And so we did RNAscope for gene A together with c-Kit, which is a marker of differentiating germ cells. And you can see the c-Kit lights up a good proportion of the germ cells within the seminiferous tubules, but importantly, does not label gene A. Gene A is a germ cell, or the cell that is labeled, shown with the arrowhead as positive for gene A, is a germ cell because it's also DDX4 positive, but it's KIT negative, which suggests it's an undifferentiated cell. And so this is really important confirmation that the identification of this gene using our single cell data has pointed us in the right direction towards a novel marker, and we're currently following up on that observation now. So just to wrap it up, I'd like to give you a few take-home messages to think about, and again, to stimulate any additional questions you may have after my presentation's finished. So I think it's clear many from my field, and I'm sure many other fields, are beginning to see single-cell RNA-seq become almost a standard technology to interrogate any biological system. And so since there are commercial platforms and academic level platforms available, these data are becoming very common, and so is then the need for validating those data. So how to do that? Well, I think at the protein level, a really good solution is to do single-cell Western blot, because that provides both the quantification and the singularity and the ability to do so with ease that is not as easy using other methods like immunofluorescence in tissue section. But let's say you're not really a protein biologist and you care much more about the mRNA. Well, you can also validate these data at the single cell level in situ using technologies like RNAscope that provides single molecule detection by fluorescence in situ hybridization that's quantifiable and, again, retains that singularity that was really the basis for doing these experiments in the first place. So I think the toolbox for taking single-cell RNA-seq to the next level is really becoming much more full, and there are technologies available that will allow you to very easily and quickly move from the discovery phase through validation and move on to what many of us are most excited about, which is to really get at the basic fundamental biology underlying our biological systems. So with that, I will thank you very much for your attention and point out first that there are people that did this work. I'd like to take credit for everything, but I didn't do this work, and it was people in the lab here in San Antonio that performed the bulk of this. So Victoria Diaz did all of our RNAscope experiments, and Shin Suzuki has done a lot of our bioinformatics experimentation. Of course, it takes others to help out, and so I should acknowledge John McCary and his postdoc, Corinne Cheng, who were instrumental in the single-cell studies that are already published. And of course, without money, we wouldn't have been able to do any of this work. And so I should acknowledge the sources of funding, in particular, the NIH, the Kleberg Foundation, who funded some of our single-cell work, and UTSA for their tremendous support of our genomics core, in which these equipment reside. So with that, thank you very much, and be happy to take your questions.
Thank you, Dr. Hermann, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, let's get started. Our first question is it possible to get a microarray-like data? Can we check a set of proteins instead of one with this technology?
Yeah, that's a good question. So the plexity of the Milo profiling for proteins is, I believe, limited to 12. And that's going to depend on a couple of factors. Number one, the kind of microarray scanner that you have available to scan the stained slides, but also the compatibility of the antibodies to be able to work together. So some of the example data I showed had one protein detected in a particular color channel, while others had more than one different proteins detected. And when you have more than one protein or antibody for that protein in a particular channel, they have to be separated by a certain amount of molecular weight. I would have to defer to the Milo technical reps to help you with the specifics. But in total, I believe 12 plex for a given slide at any particular time is the limitation. Now, I also believe that these slides, we haven't done a lot of this, so I'd really, again, have to defer to the Milo folks. But these slides can be stripped and reprobed a number of times. I've heard numbers up to eight or nine times, which means that if you multiply the number of reprobes by the plexity, you can get quite high dimensionality data with the single-cell Western blot. RNAscope, on the other hand, the standard version 2 fluorescent detection method is 4-plex. Again, this is going to depend a little bit on the kind of detection instrumentation you have. So your microscope may or may not be able to handle 4 colors at one time, in addition to the DAPI channel. So that will depend on what you have available. There is also a high-plex technology, which is 12 probe sets at a time, and that would use either 3 colors at a time or 4 colors at a time, again, depending on what you have available instrument-wise. So yeah, you can get some pretty high dimensionality data. I would not bring it to the level of microarray, but I get considerable breadth of protein or RNA assessment in a sample at a given time.
Great, thank you. All right, next question. How many proteins can be detected at the same time with this technology? Also, are there any protein sets that you custom design and provide?
So I kind of answered that first part just in my previous answer, but to get to the point about custom sets, I think, again, this is going to depend a lot on the kind of antibodies that are available for the proteins you want to probe. I believe there are a whole panel of antibodies that have been validated by Milo or customers that the representatives can point you towards. And you might want to check with the technical support people for specifics. But in general, it's been our experience that any Western blot validated antibody tends to perform pretty well using the Milo approach. So I suspect you'll be able to combine antibodies of interest based on your own experience, or if you don't have antibodies already identified, that the Milo folks can help you identify those.
Thank you. Okay, this next is a two-part question. I'll start with the first. How well are markers known for the numerous cell types in the spermatogenic lineage?
Yeah, so this is kind of the underlying problem that we have faced. There are a number of very well-known markers that are known to be expressed by particular cell types in the spermatogenic lineage, but most of them mark very broad populations, so they're really not suitable for nailing down the biology of specific subsets of spermatogenic cells. So that's why we undertook this set of experiments to identify markers that would allow us to resolve at much more high resolution the particular cells that we're interested in, and in particular here the spermatogonial stems.
Great. The second part of this question was, did you uncover any potentially novel phenotypic markers in your sequencing experiments that you were able to validate on Milo or RNAscope?
Yeah, absolutely. In fact, we identified so many that we haven't been able to follow up on all of them. But I did show some of our preliminary data for gene A, which is a protein that, or encodes a protein that has previously not been reported to be expressed by the stem cell population, but which we are now following up on in more depth based on the results from our RNAscope experiments that show it is localized at the RNA level, at least, to a small subset of undifferentiated spermatogonia. And based on co-localization studies that are really still ongoing, it looks like it's restricted to the right population. So, this is a great example of where we could not have validated that particular marker using a protein assay because there aren't good antibodies for it. So our strategy to go after the RNA in situ was very helpful.
Great, thank you. How do you validate an AB on Milo, and do you have a positive control or SPD?
So, a couple of things come to mind here. I think obviously validating antibodies is a tricky business in any kind of assay, and I always tell folks in my lab to use the best positive and negative controls they can. So if there are known cell populations that you know don't express the protein of interest, certainly include those in an experiment. You can do side-by-side or settle cells that are positive for that marker on one side of a chip and cells that are negative on the other side of the chip. It's not perfect, but it will give you a sense for whether that antibody does perform in the Milo experiments. Standards, so molecular weight standards, you could load normalized known proteins that will take away some of your ability to use all the channels, but that's certainly a possibility. It's something that we asked when we started doing these experiments. Could we get a better measure of the molecular weight of the detected proteins? And it's really kind of tricky. So I would say it's not something that we do routinely, especially for very well characterized proteins, but it is possible.
Thank you. Another two-part question. How easy are the single-cell Western and RNAscope workflows to perform, and how much assay optimization was required for your samples?
So I'm a PI, so I sit in an office all day, and so easy is much different for me than it is for folks actually doing the work. So my answer may be a little bit biased. I think that these are very easy technologies, both the Milo and RNAscope, to utilized by labs that haven't gone down that road before. We've done some optimization for Milo. So that actually surprisingly worked very well on our first shot, and we simply had to settle on an appropriate settling time, and that would be suitable for our cell types, so that we got the right number of wells filled with cells. But besides that, I think having high quality antibodies is the biggest barrier. So, if you have antibodies that are very well characterized, that you have good experience using, then more than likely, I would say you'll have pretty good success with the Milo. We do use this instrument as a part of a core facility, and we have had customers that have struggled with particular antibodies of interest, but we've been able to point them in the direction of some alternatives that have worked better for them and given them good data. So that, I think, for Milo has been pretty easy to adopt for most people. RNAscope is equally robust. So when we started using RNAscope for in situ hybridizations, we had been struggling for several months to get alternative technologies working without any luck. And on the first try, we followed all of the guides for preparing tissues, for sectioning the tissues to enhance the likelihood of success. And I should say ACD has done a fantastic job of establishing those protocols. So go on their website and download them and read them. And if you follow their guidelines, you will have the kind of success we did, which was just extraordinary. We have not yet had a probe that hasn't worked. And people find that hard to believe, but I'm not overselling this. It has been a remarkable success for us.
Great, thank you. Okay, next question. Looking at the transcript and their gene products, do you generally see a strong quantitative correlation between protein expression and mRNA across all cell types you looked at?
This is a question, the answer is really dependent on the stage. So in our experience in cells with the testis, within the testis, cells at particular spermatogenic stages, really phases, stages, a different component of our system. So at different points along their developmental progression, have different susceptibility to post-transcriptional regulation, and we know where those are. And so there are some genes that we've looked at that do not correlate very well at the protein and mRNA level, and there are others that are highly correlated. So it really depends on the stage at which you're looking in the spermatogenic lineage. Now, how to go back and address the discrepancy, I think, is really system dependent. So for us, an example is a gene that encodes a receptor tyrosine kinase KIT. That particular gene we know is expressed at the mRNA level, and there have been a number of studies that have shown that in the undifferentiated spermatogonia, but that don't turn on at the protein level until those cells see retinoic acid to induce their differentiation. And there have been studies that have gone back and shown that treatment actually, or that exposure actually forces the mRNA to the polysome fraction and enhances its translation. So that's a difficult question to address, and it's really highly dependent. But in cases where the mRNA and protein do directly correlate, then that's an easy output.
Great, thank you. It looks like we have time for one more question. What kind of sample preparation protocols do you use to get single-cell suspensions of your samples for downstream single-cell Western analysis?
So just about all of our experiments start with tissue. So we will take testicular tissue and use enzymes to digest that tissue into a single cell suspension. Most of the time that involves a tryptic enzyme to cleave protein, so we'll use trypsin, and generally tissues don't like that, so we often include some DNase to digest any DNA that arises from lysed cells. In some cases, we have to add collagenase enzymes to digest basal membrane proteins. And invariably, we have to strain our cell suspensions to get rid of debris and clumps of cells. And depending on what we're doing, we may do that more rigorously than others. So down to a 35 micron strainer or something like that. But in general, that seems to work well for both the single-cell RNA-seq and the Milo. And of course, for RNAscope, we don't need to do that because it's the intact tissue.
Thank you again, Dr. Hermann, for your time today and your important research. We would also like to thank LabRoots. Thank you. We would also like to thank LabRoots and our sponsor, Advanced Cell Diagnostics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.